So thank you, Matthew, and thank you, Simit, for the organization. I know that it's a lot of work, so it's nice to be here together, and thanks for the invitation. So the title was Multiscale Phenotyping for in a diversity of climatic scenario, but I've really loved the introduction of the session, which is adding value to phenotypic data. I thought it was really great, so I have added to my own title. Right. Okay, so is it better? So before I forget, so this work involved INRA, the Phenom project, that is the French infrastructure, a European project that was the IPPN, and the two European projects that are drops on Amazing, in which part of you have worked together. Right, so we all want to find QTLs of tolerance, whatever it means, and either from a platform or from a network of fields. And the real questions are where, when the alleles are positive, is it reproducible, what can I do with a platform, with a field, etc. So what I'm going to present is basically a combination of phenotyping and variotyping and modeling, transcale. Many things I will say are actually very similar to what Fred has just said, just we don't tell them in the same way. Right, so one of the key questions, I think, transpiration, photosynthesis, vary at the time scale of minutes. So whoever has measured gas exchange, we know that it can be completely different at 1 minute distance. And the plant environment varies at this time scale. So we are more or less used to have a variability of climate over years. We have basically the same over days, but if we take a single day, the plant has huge variation of environmental condition between one day, and it shows. So we can have a plant here, they are completely happy in the morning, and 6 hours later they are completely stressed. So the plant is continuously adapting. For growth, it's basically the same. So if we have here night, day, night, so we have the light like that, transpiration like that, leaf elongation rate that is maximum during the night, in half an hour, boom, goes down extremely rapidly. So everything is changing all the time, between hours and between days, with time frames of about 20 minutes. So it's something that really changed, the environmental change, everything changed. So we want to phenotype yield, and this is over a month, and we have mechanisms that are over minutes. So there is a trouble here. And one of the trouble is that the optimum for each mechanism depends on the long-term scenario. So we can have mechanisms that have contrasting effects on yield depending on scenarios. And sorry for those who have seen that several times. But if we take, for instance, closing stomata, increasing water use efficiency, decreasing leaf area, it's definitely positive whenever we have a terminal stress, and it is definitely negative if we have water. So it's stupid to have kept water for later if we are going to have more water. So the same trait is either positive or negative depending on the scenario. And sorry for the root lobbying, but the root system is just about the same. It's a very good idea to have a deep root system, to increase the root system if we have deep soil or weak soil. It has been shown several times that if you have a shallow soil, you had better to put your carbon elsewhere than in the roots. So again, the same trait can be positive or negative. So we have a tension between genetic variability of mechanisms, organ, minutes, and yield, canopy, month. So that's the reason why probably if we talk of phenotyping, we need to have different types of phenotyping platforms. We want to have high precision platform to study things, cell and organ at a time course of minutes. At the other extreme, we want to have a multi-environmental network, like Fred has just talked, in which we have canopies in a range of environment, and we are talking of months, and we probably want to have something intermediate. So that's the reason why in Phenome and Emphasis we are trying to develop these different type of platforms. The typical things that we can see in this platform are different. So the typical mechanisms here, we can do hydraulics, metabolism, signaling, this sort of things. Here we can have things like light interception, water transfer, etc. And in the network of fields, we cannot expect really to have mechanism. We'll see overruling emergent properties, trades off, etc. But we will never have a real decent mechanism in the field. But it's still useful to do the field. So there have been plenty of reviews claiming that it's great to be here because we have a good, what I call the ratio of biology to other processes. Don't ask me how I calculate that, but I think it's intuitive. So we can have biological mechanisms here and more difficulty here. And the relevance for field is good here and quite bad here. So we can talk for the rest of the day whether it's better to have one or another. What I'll try to show that the added value is to do the transcale analysis because we need all of that and to travel. So I will try to show some example of traveling. Right, so I will begin with this, guys. So field multi-environmental network in canopies, etc. And we are asking exactly the question that Fred was asking, and he's participating to this work. It's where each combination of allele is favorable. So we did in DROPS, European project, and its continuation in Amazing, and with the help of many companies, breeding companies, and to Fred and Alain Charcosset, we had 16 fields, 2 years, 2 watering treatments, so a decent number of experiments. And we did exactly the type of phenotyping that the breeders do already. So nothing new, only yield component, that's it. The only thing is that we asked to have a decent environmental characterization. So this costs, if we talk of a cheap phenotyping, this costs about 300 euros, so it's decent. But it's work. But I think you will see that it's helpful. So we had the 252 genotypes. That's an old slide, so we have now more than 8,000 markers, and we had yield variation from 5 to 12 tons, depending on the sites. So, for instance, 3 sites here. So we have this is the genetic variation within our panel of genotypes in 3 sites, and you can see that the yields are quite different between sites. And as everybody else, when we did GWAS in each site, we had a lot of significant SNPs that moved all over the place, and it was very difficult to make any sense of that as everybody else. So the added value, we wonder whether if we classify the environments, we can make some order out of this mess. So what we did was, actually we did not take these environments to do our environmental classification, but we took actually 40 sites, 35 years, and we ran the APSIM model over the whole stuff. So this was a work with Graham Hammer, and so we could identify some essentially 6 big scenarios of temperature at flowering time and soil water potential. And what we did afterwards was to reorganize our experiments. Now, these are our experiments out of these big scenarios that do represent whole Europe. And we have some series of experiments in which we have cool temperature, hot during the day, hot during both day and night, well water not that bad, etc., etc. So I will, for this talk, say just well water and water deficit, and this will be it. So what happens if we classify our environments in this way? For instance, one peak can have very positive allelic effect in one type of environment and nearly no effect in another one. So that was part of the game. We all know that, but the interest of that is that we can organize our data allowing this type of analysis. So this is for one QTL, but actually each of our QTL have its own story. So for instance, this guy is is really negative in this type of environment and positive here. All these guys are good in mild water deficit and bad in well water, etc., etc. And we can even regress the allelic effect here against one condition. Here it's soil water potential as it was measured in all our fields. And you can see one QTL whose effect decreases with soil water potential. So this is dry, this is wet. Where this QTL actually is rather negative in well-watered and quite positive in drought. So we have that for each of our QTLs. And so this can help to understand what happens. Can we identify genes out of that? One interesting thing of this is GWAS, so we have about 12 genes about in our interval of confidence. So in some cases there are some miracles. So we have one QTL that is positive in water deficit, negative in mild water deficit or well-watered. So we know that this QTL increases the hormone abscisic acid, and it included the gene for abscisic acid. We exactly expect these sort of things. So we have a coincidence between the potential gene and its different effects in different environments. We have to say that this is the exception, because most often we have absolutely no clue of the effect of any of the genes in the QTL. So, what is the added value here? So we have a classical experiment, simple phenotyping. The only thing is envirotyping based on on-field measurements. And the added value is that from a lot of non-repeatable SNPs, we got 48 QTLs, quite good, scenario specific. So each of it has different effects. And in total, they capture about 80% of G and nearly 70% of G by scenario, so it's decent. And scenario specificity helps getting the candidate genes, but again, essentially, we have no idea of the mechanisms. So we can try to have something slightly different still in the field, which is to have a continuous approach in which we use relation with environmental variables. So I will give just one example. The field that were in well-water conditions, so soil water potential above 0.1 megapascal. And what we had is we have modeled light-intercepted pre-flowering with APSIM, so it's here. And we have plotted grain number in front of it, and we see quite a nice regression. So each point is a field, and we have it's as if it was different different treatments in one big experiment, and we have a nice relationship for one genotype. And now, if we look at all the genotypes, we have a difference in slopes, quite large, with a good heritability, and we calculate the QTL for this slope. So we have QTL of efficiency. We don't know exactly what it is, but we can predict the effect of so we can predict the effect of light and the genetic variability of that. Right. So here the added value, I think, of regression analysis is we can relate differences between sites to physiological variables, and we have QTL of slopes that handle the G by E. But what are the mechanisms? We don't know. Is it photosynthesis? Is it light interception, etc.? So I think we cannot really do that in the field, and that's why we have also a high throughput platform. And this is exactly the platform that Jorens has presented this morning, so I don't present it again. And so this equation was presented as well. So we decompose biomass in incident light interception, and radiation use efficiency. So radiation use efficiency is a proxy for photosynthesis. And we try to do that in the platform with exactly all the set of methods that Jorens has presented. So first we have to deal with the local available light, because we know that each plant has a different light, so we take care of that. In addition, part of the interception is environmental, because you have some plants that don't compete with any other because they are at the limits, they are outside, so we take care of that. But the important thing is this. And we can give a first conclusion for this panel. The conclusion is panel dependent. It can be different in a different panel, but basically light interception was a major player for biomass. So if you have here corrected intercepted light and measured biomass, the correlation is not perfect, but if you see the correlation with radiation use efficiency, you would see something like that. Right, and inside that, the leaf area, so just leaf growth, is really the major player for interception. So, of course, we have architecture, etc. But if we look here, the leaf area per plant and intercepted per plant, at the beginning it correlates very well. So this is time. So you have a perfect correlation, and only at the end you have some deviation that is linked to the plant architecture. So, to make the story short, we can calculate something that we call competitiveness. Don't ask me exactly, well, we'll have time to. So, but basically, this plant is less competitive than this one. This is more able to capture light, even in the field. And you can see that if we regress leaf area here and intercepted light, we have the same graph that I showed before, but it's better to be like that. So we are at the top of the cloud than like that. So finally we can dissect this Monteith equation. Okay, incident light, so we are not interested in that. But we have leaf area, and we have in the panel quite decent heritabilities. Architecture, we have acceptable heritabilities, so we have QTL for all of that. Radiation use efficiency. So there was a question this morning about the genetic variability of photosynthesis. In the panel that Jorens presented this morning, that were lines, tropical lines, there was a very special genetic variability. In this one, it's very small, if any. So we have QTLs, but are they imaginary or are they good? We have still to work a little bit on that. Right, so but the take home is that the major effect on biomass was leaf area and leaf growth. So the important thing, so if we want to connect, is that every term can be inserted in a crop model, because a crop model has exactly this term, so we can decompose these three terms, and put the QTLs exactly in the way as Fred has shown, and we can predict our genotypes in different type of sites. So now if we compare what I said before, so we have in the field this apparent efficiency, so it was very tempting to say, okay, that's photosynthesis. Actually, it's probably not photosynthesis, it's probably the ability to capture water, light, that was the major genetic difference. Again, in a different panel, it might be different, because this is the genetic variability we have in this panel, although we have seen the same in other panels. So the added value here is we have QTLs of efficiency of light in network of fields, and we can say, in this case, going to the platform, that is probably linked to leaf growth, interception, or all a category of gene that is linked to growth rather than photosynthesis. So, if leaf growth is important, we might like to study it in more detail, so I will do that rapidly in different type of platforms. So we have a type of platform in which we can measure growth every 3 minutes if we like. So we can have traces like that in bright days, night maximum, day minimum, and you have in cloudy days, it's less different. So for a given genotype, we can put all these points together, and we end up with a reaction norm, response curve, whatever, between air dryness, vapor pressure deficit, so the air is dry here and wet here, and leaf elongation rate. And because we have different genotypes, with each of them has its own slope, so we can make QTL of this slope and play with the QTLs as I've presented. So this is with this platform, but with the other platform I don't present it, and with the platform I've just presented, we can do basically the same thing as well, at whole plant level. Right, so leaves only probably have limited effect. So I just wanted to present one thing, that a silk, for those who know maize, silk is an organ that emerges from the ear and collects pollen, and it works basically as a leaf. So the title is The Silk is Another Leaf, but it oscillates between a maximum during nights, minimum during days, and oscillates like that. If you increase the VPD, the oscillation are greater, etc., etc. So they behave hydraulically very similarly to a leaf. I want you to see this. So we have here a plant in water deficit and a plant in well watered. So you see after 3 days, you see the silks that stop growing while they continue growing in well water. And this is probably linked to the cumulative growth they had before. And so we have something that is linked to growth. Is it important for yield? The answer is in our hands, yes. So we have here the final number of emerged silks. So this is done 3 days after silking, extremely early, and correlates really quite well with the grain number. So this is just to say that it's important. The growth has an effect even for the reproductive development. I will skip that, and this is just to say that we do have a good genetic correlation between sensitivity of leaf growth and sensitivity of silk growth. These are near isogenic line that we tested for both traits, and you can see that it's not that bad. So perhaps and that's important for phenotyping, when we measure a trait, we actually can measure another one and keep the same genetic architecture. And this perhaps explains, to our surprise, so when we did with it was with Limagrin, so a network, we have the sensitivity in the platform and in the field, calculated as I said before, and it works decently well. So the final question, and here we are back to modeling. So we know that we have a genetic variability we have genotypes that are stable, continue growth at quite low water potentials. Others are very sensitive. Is it better to be sensitive, or is it better to be resilient? The answer is it depends, as you can imagine. But we have to explain this, it depends. So, So we use the modeling, so we took APSIM, and with Graham we changed a little bit to have explicit growth and the responses that I've shown, and we simulate that in whole Europe. So we just I present here the difference between a stable and a sensitive genotype. So here you have a color code in which it can be either positive or negative. So you can see, it's a mess, exactly as in experiments. But now if we really study the output of our model, it becomes completely understandable, and it's exactly what you can say. So if you have a severe water deficit, it's better to be sensitive because you keep water, and if you are in a mild water deficit or no water deficit at all, you had better to continue growth as you are. So it's another way to say any trait, blah blah blah, can be positive, negative, etc. But I think with the models we can assess, without having all the genetic architecture of the trait in the model, we can predict where it is positive and where it is negative. So the added value here, so we are here with a very detailed platform, and it's not the general case. It could go until the yield. Again, it's not a general case, but in this case, it's a hierarchical mechanism translated to yield via reproductive resilience. So we have a proxy of reproductive resilience with leaf growth, and I think it's an interesting idea to keep, because it's very difficult to phenotype reproductive growth. And we have a precise idea about where and when this mechanism can be favorable. So, conclusion. So, probably everything that is yield has to be in fields, in networks of fields. There is no way of having decent yield in phenotyping platform. They are not for that. So, we can do that with very simple phenotyping, but we absolutely need the environmental conditions. This is exactly what Fred said 5 years ago. And I had proper statistical designs, and Fred said about the row column models, etc. It's essential. Are we really wanting to have phenomobile or UAVs in this type of big networks? To be honest, I don't know. But our experience is perhaps is not that trivial, even with drones, to run drones all over the place. So probably we need other type of platforms. Can be in the field, can be in the greenhouse, but that are for identifying mechanism, disentangling the genetic variability, etc., for having the transient short-term mechanism, for having traits that cannot be measured in the field. I've shown that. And I want to finish with this idea. Okay, phenotyping platforms do not represent the field. It would be silly to, but they are for something else. But remember that a field does not represent another field either. So the question of, is my phenotyping platform representing the field? The answer is no, but it can help in understanding how different fields are different. So probably we'll have to live with a diversity of platforms. I've tried to show that they can be useful if we have the added value of combining scales, but provided that we have a consistent environmental characterization and allowing trans-scale modeling. And thank you for all participants, so all the people who did the experiment, all these companies, people who measured traits, genetic analysis, and all the people of Lech who participated to that.